Good afternoon, and thank you for having me. My name's Lynne Cox. I'm a biochemist at the University of Oxford, and I've been working on aging for the past 28 or so years. I'm not an eye specialist, but what I hope that I can bring from the aging field is some new insights into how we can understand and treat macular degeneration. So my talk today is about developing new treatments for AMD by targeting underlying aging processes. But let's start with aging. Now, everybody knows what aging is and what it looks like. I mean, we're all familiar. I can see it in the mirror every day. But how does it impact us as people, and how does it impact the eye in particular? And if we look at what happens as we age normally and healthily, if we take a person who is 30 years old as a baseline and look at them when they're 75 years old and they're happy, healthy, no underlying conditions, their body has still changed. So nerves are going more slowly. The heart is down to less than 3/4 of its ability to function that it did in a 30-year-old. Kidneys down to about 70%. It gets a bit more worrying when we start looking at things like the brain. So the brain shrinks by half by the time you're 75. And I'm well on the way to that, and it's slightly concerning. Vital capacity, the amount you can breathe in and out, again, down to half, and hand grip strength down to half. And that in itself is one of the biggest single predictors of mortality that we have, which is, again, slightly concerning. But then the real red flags come up. And remember, this is normal aging, not age-related disease. The amount of oxygen you take up on exercise once you hit 75 happy and healthy is only down to 40% of what it was when you're younger. Taste buds, a third of what they were, which is why food just doesn't taste like it used to, or why people are reaching for the salt on a regular basis. And perhaps one of the most worrying things is our ability to rebalance after we've stressed our bodies really goes down. So, for example, the pH of your blood, the acidity changes when you go, say, for a run. You're catching a bus, so you have to speed up a bit. When you're young, it comes back to normal quite quickly. As you're older, it takes forever to return to normal. And that's what we call loss of homeostasis. And it's actually not very healthy. Yet this is in normal, healthy aging. And of course, all these changes affect not just the heart and the nervous system, they affect the eye as well. So there are all sorts of changes that I'm sure a lot of people are aware of. Lower tear production, so eyes get a bit dry. The proteins in the lens start to change, which can lead to cataracts or long-sightedness. So many people need reading glasses over the age of 40. The vitreous humor, the gloopy stuff in your eye, starts to shrink, and that causes problems with the retina and also seeing floaters around your eyes. The ability to distinguish between similar colors is diminished, and there's more difficulty adjusting to light levels, so changes in light levels, which can also lead to problems with night vision. So a lot of older people find driving at night difficult. Now, how are we going to start understanding this? Well, we know that aging starts at the level of the very building blocks of the body, the cells of the body. So if we think of the body as made of Lego, we all know what Lego looks like. There are about 200 different types of Lego, all different shapes and colors and sizes, 200 different types of cells in the body, and they all go together. We get 37 trillion different Lego pieces to make up a person. And that person has, for example, 2 billion heart cells, 2 billion muscle cells, 35 billion skin cells, 50 billion fat cells. And in the eye, in each eye, there are around 7 million cones, which allow you to see in color and daylight vision, and between 75 and 150 million rod cells, which allow night vision. So we are made up of many cells. And of course, if something goes wrong with the cells, that backs up and makes things go wrong with the whole body. We can look at aging cells in the lab, and this is someone in my own laboratory culturing human cells in little dishes. And on the So that's the person on the left-hand side, if you can see that. On the right-hand side, there's a picture of a digital microscope with a small flask of human skin cells on the microscope, and the picture on the screen shows those skin cells magnified quite significantly. So we can grow human cells in the lab, we can manipulate them, we can do all sorts of things with them to understand how they're aging. So stepping back a little bit to macular degeneration, we know some of the causes of macular degeneration. One of the biggest things, of course, is simply the passage of time. I've got a clock and a timer on the right-hand side of the screen, and we all know that old Father Time keeps ticking along and things can really change over time. But there are some things that are actually major triggers for premature aging and AMD, and that includes smoking and exposure to blue lights. So of course, I'm currently looking at a screen. My eyes are being exposed to blue light all the time. And we don't know what that holds for the future for children growing up with blue light exposure. But what we do know is that all of these things can trigger a process of cell aging called cell senescence. So senescent cells cause aging. We know that the number of senescent cells or aging cells goes up as we get older. We know that removing senescent cells improves health, at least if you're a mouse. And we know that if we inject senescent cells into young mice, they get old prematurely. So these things, these normal body cells that have got old, drive aging. How do they do that? Well, in the middle of this picture is a cartoon of a senescent cell. And there are many things wrong with it. First of all, it can no longer repair and replenish. So older people suffer from poor wound healing because their cells don't divide as often as young cells, so you can't repair. They accumulate a lot of damage to the blueprint of the cell, the DNA that codes for all of the processes that are happening in the cell. And then because of these changes in the DNA, it leads to changes in expression of our crucial genes. So sometimes genes need to be switched on and off, and the off is actually as important as the on. But in aging, sometimes that gene expression is kept on. It's like having a tap turned on in your bathroom the whole time. And of course, you know what happens if you leave the bath taps running, you flood the bathroom. So this is not good. And then these cells that are really quite damaged stay alive. Normally damaged cells would die, but these stay alive. They're like zombies. They're falling apart, but they're still sitting there in your body. And they fail to take out the rubbish. So they're full of gunk. They clog up with all sorts of damaged features in the cell, damaged proteins, damaged lipids, and they fail to do this recycling. It's called autophagy. And they also harm their environment. So they're kicking out a pile of inflammatory mediators, enzymes that chew up your tissue, hence the wrinkles around your eyes, because these are enzymes that are eating into the structures that support your skin. And then factors like VEGF, which a lot of you, I'm afraid, with AMD will be familiar with, because it leads to formation of new blood vessels. So the inflammation is like lighting little fires around our bodies, and then you have a formation of multiple new blood vessels in wet AMD. So senescent cells are harmful and they can cause aging. And they may be involved in AMD. So if we look at dry macular degeneration, which, as you know, accounts for between 80 and 90% of cases, there is a major failure to recycle worn-out molecules, and this can lead to deposits called drusen, proteins that just sit around and aren't removed from the cells. There's an accumulation of DNA damage and inflammation, and tissue damage and breakdown as a consequence. So these are all features of cell senescence, and they're features of dry AMD. Now, in wet AMD, fewer cases, so 10 to 20% of people with AMD will have wet AMD, but it is one of the biggest contributors to failure of vision. So it's something that definitely needs seeing to as quickly as possible. And the reason that it can cause blindness is that new blood vessels can grow across the back of the eye, and they can actually block the nerves that are needed to see, to convey that message of sight from the eye to the brain. And also, the new blood vessels can leak into the eye, and they can displace the back of the eye, the retina. So we need to do something about it, and senescence may be involved. So what can we learn from senescence biology to help us treat AMD? There is good news. There's been a lot of very good research happening over the past decade and accelerating now that shows that there are ways that AMD can be treated now and perhaps even more effectively into the future. So what are these new, current and new treatments that target senescence? So on the slide, I have a cartoon. On the left-hand side, there is a picture that looks almost like a hand. It's a small retinal pigment epithelial cell, RPE, and those cells are very, very important at the back of the eye. Now, over time, these cells can become old and senescent and very different in the way they function, and they start throwing out other molecules. So there's some stars emerging from the top of that cell, which are the inflammatory factors and all the things that can drive blood vessel formation. So ways that we can deal with this is maybe when the young cells, the young functional cells are worn out, perhaps we can replace them with more young functional cells. Or we can delay the time it takes for the RPE cells to get old and dysfunctional. Alternatively, we can try and block all the nasty chemicals that they're kicking out. Or if they're that bad, we could simply just try removing them. And maybe the other cells that are still functional will take over, and that will solve the problem. So there are 4 different ways that we can think of targeting senescence in AMD: replacing cells, delaying the aging process, blocking the harmful chemicals, or removing the cells. So if we look at these step by step, if we look at replacing the damaged retinal epithelium, so the layer at the back of your eye that helps you to see, this might be a solution. Now, it's a bit science fiction, but it's now science reality. It is possible to take cells from patients, just normal blood cells, and turn them into something called stem cells in the lab. These are called induced pluripotent stem cells, lots of scientific terms, I'm afraid. But they go through this stem cell period where you can then persuade them to turn into an RPE cell. So those very important cells at the back of the eye that help you to see. And you can do this in the lab. You can grow these cells and you can line them up on a scaffold, so essentially like a plaster. So you put cells, human cells, in this plaster, and those patches can be surgically implanted into the eye. So the picture at the bottom shows the back of the eye in blue with a patch in green and red and yellow that's been implanted into the back of the eye and replaces the missing cells, the cells that no longer function. And these patches have been shown to work very well in rats and pigs. And they worked so well that they're now in clinical trials in humans in the USA. So imagine it's you cut yourself, you put a plaster on the cut, your eye stops working. It's slightly more complicated than a simple plaster, but you essentially insert a plaster at the back of the eye to patch it up, and that plaster contains the cells that can integrate and start working. So very, very exciting new ways of thinking about macular degeneration. We can also think about delaying the onset of this aging process. Now, what we know is the chromosomes that package up our DNA, the blueprint of our cells, they have ends that get shorter as we get older. So there's a cartoon showing a young, fit, healthy chromosome helping an old, sad chromosome up the stairs that has short telomeres. So these chromosome ends, telomeres, get shorter with aging, and there are and we know that that's a trigger of cell senescence. But what we also know is there are some drugs that can keep the telomeres longer. And a particular drug called TA65 was tested in a clinical trial, a small trial with 38 patients. And over 6 months to a year, keeping the telomeres, the ends of the chromosomes, longer showed macular function improvement that was maintained for a whole year compared with placebo treatment. This is obviously still very early days, but it's quite exciting. And then we can think about blocking those nasty chemicals that come out, blocking inflammation that's lighting all those little fires that are harmful for your cells and your tissues. Now, there's a very complicated diagram on the left of the back of the eye, but essentially it's showing the stages of progression from a healthy eye to people at risk of AMD, to the back of the eye when it's actually already developed macular degeneration. And along that progression, there are some chemicals that are really important in driving inflammation. And these have a strange name. They're called complement factors. And there's complement C3 that's highlighted in red, and then complement C5 that's highlighted in blue. Targeting those with drugs that blocks them from working can reduce progression of dry AMD by between 18 and 35%. And of course, this is a massive new advance because until very recently, there were no treatments for dry AMD. Now, the drugs have horribly complicated names. I can't say them properly, so I'll just leave you to read them. But pegcetacalopan and then avastin Captad, I told you I couldn't say them properly. These are drugs that are new to the market and are showing great promise in dry AMD. But obviously, they're not a cure-all. It's reducing progression 18 to 35%, but that's better than we've ever had before. And then there's other factors that are important to block. So in wet AMD, it's the formation of the new blood vessels that causes the most problem. And those blood vessels respond to factors, particularly vascular endothelial growth factor, which helps new blood vessels to form. So blocking that VEGF, as it's called, is a way to block the formation of new blood vessels in the back of the eye. And injections of anti-VEGF antibodies can reduce vision loss by 50 to 70%. And I presume a number of people listening to this right now will have direct experience of those eye injections. There's multiple different drugs on the market, Avastin, Lucentis, Eylea, Beovu. And what these drugs do is not just halt the progression, but potentially they can also improve vision. So if we take a vision chart and we work out how far up the vision chart someone can see or how far down, and then look over time at patients with AMD who didn't have the treatment, they had placebo, they could see 11 fewer letters on the chart over time. Whereas with treatment with Lucentis, they could actually see 6 more letters than when they originally started in the trial. So not only does it prevent progression, it can actually lead to improvement. Now, I have no financial interest in Lucentis or any of these companies, but I think it's absolutely fascinating that biology has taught us what we need to target, and then companies have come up with drugs that are effective. Then we can look at removing senescent cells to improve AMD, and we can do this in the lab. So I've shown some blue cells. This is the DNA stained up in the cells in non-AMD. And then in an AMD model, these cells become senescent, and we can mark them with dyes. So here they're shown in pink. And after treating by removing senescent cells, the majority of the cells now are blue again. They're healthy, reasonably normal cells. So we can get rid of senescent cells in retina in the lab. Can we do it in vivo, in a living eye? And the answer is yes. If we look at the back of the eye, the retina in mice, it has beautiful separate layers that you can see down the microscope. The picture at the bottom has black and pink and purple layers, which is this normal structure of the eye. If we look in a macular degeneration model in the mouse, those layers are very disrupted. But treating the mice with a drug that removes senescent cells allows the eye to heal and restore those layers that are important for vision. So in conclusion, cell senescence is a potential target for age-related macular degeneration in many different ways. We can replace the damaged retina with these stem cell patches. We can delay the onset of senescence, potentially with things that help your chromosome ends stay intact. We can block inflammation through the complement pathway, and we can block new blood vessel growth through anti-VEGF antibodies. And we're now looking at removing senescent cells as a potential positive treatment that works in mice and is now moving towards clinical trials in humans, at the moment for diabetic retinopathy, but potentially for AMD. Now, of course, many of these new approaches are experimental, and I'm not saying that people should try them themselves. It's always very important to take professional advice from someone who knows you and your eye condition. But I would like to say that it's looking a lot more optimistic in terms of treatments for AMD. So thank you for your attention. I'm going to finish there, and I'm happy to take questions.